Hello guys, welcome back again. In this video we are going to see somatic embryogenesis, and we are going to see this process, steps, procedure, and what's the importance does it have. So let's start this slide. So first slide is coming through. Let's see what we are going to study in this video. First one is intro, and second one is we are going to study in this video is types of somatic embryogenesis. After that, development stage, factors that will affect somatic embryogenesis, what's the importance does it have, conclusion, and this all these things we are going to study in this video. Let's come to the first part, that is your introduction. Now, what's the meaning of somatic embryogenesis? It comes to your mind when you see this thing. Then the process of regeneration of embryo, from somatic cells, tissue, organ is regarded as somatic embryogenesis. It is like a asexual reproduction. It doesn't have any sexual object in it while it is creating a embryo, so you can call it somatic embryogenesis, or you can say embryo is generating from somatic cells. Opposite of zygotic or sexual embryogenesis, embryo-like structure which can develop into whole plant in a way that is similar to zygotic embryo are formed from somatic cells. Now the embryo will remain the same as we have created from our sexual reproduction. Now we have created this embryo by using somatic cells, not any sexual object. That's why we call it somatic embryogenesis. This is intro. Now come to the second slide, that is history. Now talk about the history. First recognized by the scientists Stewart and Reinwald in culture of Decus carota. Okay. Now first these scientists recovered this process in culture of Decus carota. In this, 4 different stages are there. First one is initiation, second one is proliferation, third one is maturation, and fourth one is germination stage. These 4 stages will be happening while we study somatic embryogenesis. So if you come to the development stage, now embryo will create a globular like structure. After that it will create heart like structure. After that it will create a torpedo, and at last it will create a cotyledon like embryo. Okay, so these are the step by step a embryo will be generating in this process. Okay, now come to the next slide, that is various term from 4 non-zygotic embryos. First one is adventitious embryo, somatic embryo arising directly from other organs or embryo. We can call it adventitious embryo. Second one is parthenogenetic embryo formed by the unfertilized egg. Okay, unfertilized egg, if it is creating a embryo, we can call it parthenogenetic embryos. Now what's the androgenetic embryo? This can be formed by the male gametophyte part. What male gametophyte will also create androgenetic embryo in the term of non-zygotic embryos. Okay, now come to the next slide, that one is somatic embryogenesis types. Now it's have 2 types. First one is direct somatic embryogenesis. In this process, embryo will be creating without creating any callus formation. So in this process no callus will be formed, while in indirect somatic embryogenesis a callus will be formed. Okay, so that's the main basic difference between these types. Now come to the first one is direct embryogenesis. In this, somatic embryos develop directly on excised plant without undergoing callus formation. So in this process no callus will be formed. It referred to as somatic embryogenesis. It is very rare, and the characteristics feature of direct embryogenesis avoiding the possibility of introducing soma-clonal variation in the propagated plant. It want to say to you that it doesn't create or it doesn't add any variation to the embryo. So we are not going to see any variation in the progeny plant. It will remain the same as their parental plant. Okay, now come to the next slide, that is your indirect somatic genesis. And in this process a callus will be formed and your soma clonal variation will also be added in this process. Okay, that's why it is very common, because we all want to create some advantage or new character into the progeny. Now we are going to see the steps of this all functions. Now this is your mature plant and we are going to use its only somatic object, not any sexual object. Okay, so we have excised the root cells and we are going to culture them, and this is the place we are going to culture them, and this is culture cells in culture divide to form embryoid bodies. Okay, now we have discussed the stage: torpedo, globular, and heart like stage, and in the last they will create a cotyledon stage. Okay, so first one it is creating torpedo stage and last one is cotyledon stage is planted, and it will generate a small plantlet. Okay, small form of plant will be called as plantlet. Okay, now it will if you are going to plantation that plantlet into any field, it will create a mature plant like just like their parents. Okay, so this is indirect somatic embryogenesis, because in this process they are creating a callus and we are going to use any somatic object. That's why we call it somatic embryogenesis, as I have discussed it earlier. Come to the next slide. Now come to the plant hormone that will initiate your somatic genesis. First one is auxin, and auxin will require only for induction. Okay, auxin required for induction. 2,4-D mostly used development. During the process of development, the culture media should be free from auxin. Auxin must be removed from for embryo development. So auxin is only required for embryo initiation, while development procedure this is not required. Okay, maturation required. Complete maturation with apical meristem, radical and cotyledon will be used as explant. Come to the next slide. This will be your development patterns. As you can see here, first your embryo will like this globular shape, after that heart stage embryo, and after that torpedo, and in last it will look like a mature cotyledon embryo, and you can plant it into field. Okay, so this is influenced by several factors. This whole procedure is influenced by these factors: explant, genotype, growth regulator, and nitrogen salts. Now come to one by one. First one is explant. Your explant should be meristematic portion of the plant, like immature zygotic embryo, best explant for Relacitrant species. Petiole section from youngest fully expanded leaves, establishment of embryonic culture of alpha. So a meristematic new plant can be used as explant. Come to the next slide. That one is genotype. Explant genotype has a marked influence on somatic embryogenesis regeneration. A strong genotypic effect have been shown in many species like alfalfa, wheat, maize, rice. In case of wheat, chromosome 4b is important in regeneration. So a strong genotype will also have a suitable or appropriate somatic embryogenesis procedure. Come to the next slide, growth regulator. I have already discussed it. The auxin will required only for embryo initiation. After that you have to remove the auxin and you have to input the cytokinin, because cytokinin will be used in cotyledon formation. Come to the nitrogen source. Nitrogen plant will be used as nitrate form, okay, by nitrogen fixation procedure. This is all, don't need to remember all of these. Come to the next portion. Presence of auxin in the medium is generally essential for embryo initiation. Tissue maintained continuously in an auxin free medium do not form embryo. So you can't use continuous auxin medium into the culture media, because we only use auxin for initiation. Okay, when transferred to low auxin medium, embryogenic clumps develop into mature embryo. Clumps like it is a callus like structure will be formed. Light generally promote embryogenesis. So light will generally promote embryogenesis, and high temperature usually favorable for somatic embryogenesis. Come to the advantage and disadvantage portion. Some importance of somatic embryogenesis is raising soma clonal variation. Your progeny plant will have some variation from your parental plant. Synthesis of artificial seed also can be generated by this process. Synthesis of metabolites, clonal propagation, genetic transformation, conservation of genetic resources can be used by this process. While it also has some disadvantage, you should know high probability of mutation can be happen during this process. This method is usually rather difficult, and you have to use very material and a very efficient knowledgeable person. Losing regenerate capacity become greater with repeated subculture. Induction of embryogenesis is very difficult with many plant species. A deep dormancy often occur with somatic embryogenesis. While doing this process, a dormancy also can be happen. So this is your advantage and some of the importance of this process. I have discussed all the steps, functions, and how we can use it in our agriculture, and importance and some disadvantages of this procedure. I hope you will like the video.